just pulling up my presentation for tonight, and then we can get started. So welcome everybody, and hopefully I'll be able to give you some more information on embryo transfer tonight, and be able to answer all your questions. So tonight, the plan for tonight is I will talk about what is embryo transfer, why would we choose for embryo transfer, and then as well some options if embryo transfer doesn't work. So what is embryo transfer? Embryo transfer, so we have two things with embryo transfer. One is the donor mare, and that is a mare of which you want to have offspring. So the mare with superior genetics. And then the other one is the recipient mare, and it is the mare that will carry the foal as it was her own pregnancy, and she will raise the foal. So a recipient mare, we ideally have a young mare between 3 and 10 years of age. And we like to synchronize two recipient mares at least with the donor mare, just to enable to enhance the success, to have a good synchronization of the recipient mares. And what we mean with synchronization is that the recipient mare should ovulate should ovulate either 1 day before to 3 days after the donor mare. And what I do find very important as well as the recipient mare is her character, her temperament. You need to see a bit the recipient mare as a nanny. If you're having a nanny for your child, you want somebody that is raising your child properly with the same kind of ideas and standards as you want. And if she's a naughty mare, she will teach you for a lot of bad behavior. And I think a lot of people ask me about size as well, but as long as you have a horse, I don't think in a horse of a decent size, that's fine, as long as you don't go to the extremes of putting a pony in a real big mare. Then you should be fine, because it's at the end the genetics of the donor mare that will give the size of the foal. So if you don't have your own recipient mare, you can lease one, and the good thing is for embryos, they can be transported. And this picture is from when I was working in Argentina, and I actually used it as a little seat when I was waiting for the bus, took the embryo home, and I went to transfer it the subsequent day. So that is absolutely possible if you don't have your own recipient mares. So the first step in embryo transfer is synchronization of your donor mare and your recipient mare. And so your donor and your recipient mare need to be in season at the same time. And as I said earlier, the recipient mare needs to ovulate either 1 day before to 3 days after the donor mare. And in order to do that, we use different hormones such as prostaglandin or like luteinized or estramates to bring the mare in season. We can also make them stop to come in season, just slow them down using Regimate, and we can induce ovulation using an ovulation-inducing agent, just as such as Coralon, Deslorelin, or others. Then a very important step is donor mare management, because very important that you won't have a successful embryo transfer if your mare isn't pregnant. So when I inseminate a mare for embryo transfer, I will do the same procedures as I would do, like she would carry the embryo herself. So if she has fluid, we would treat that fluid. If I think there might be an infection, I would firstly treat that and diagnose the infection, take an endometrial swab, do a small volume lavage, something like that, just to make sure that that embryo, even though it's only going to stay for a very brief period of time in the uterus, so it arrives in a clean uterus. If she needs to have a vulvoplasty, so a Catholic procedure, if she needs to be stitched, we will stitch her up. Even though we do the flush only a week later, I would stitch her anyway. to do a successful embryo flush, it's important that you have the exact timing of ovulation. And the day of ovulation is what we call day 0, and we use that to calculate when we would do the embryo flush. And we do the embryo flush at day 7 or day 8 after ovulation. If I inseminate the mare after ovulation, so which is often the case with frozen semen, I would flush her day 8. If I have chilled semen, I do day 7, day 8, depending on schedule sometimes. Or older mares, I would tend to do rather on day 8. Why not on day 6? As you see in this up here, if we flush at day 6, we actually don't recover embryos, and the reason for that is that once ovulation occurs and then the oocyte is fertilized, the first 6 days of development happen into the oviduct. So it's only around day 6.5 roughly that the embryo arrives into the uterus. So if you flush too early, the embryo won't be there. And if we flush too late, and we will see later on as well, I will speak about maternal recognition of pregnancy. So tricking that uterus of the recipient mare of thinking, oh, I'm actually pregnant, just doesn't, there's only a certain period of time we can do that. And furthermore, if the embryos are too big, they won't fit in or straws we use to manipulate the embryos, and they become very fragile, and we are more likely to damage the embryo before we do the transfer. So it's really important that the vet that is doing the monitoring of the donor mare, that he knows, or he or she knows exactly when the mare has ovulated, and we use for that certain drugs such as Corlon and Daslorelin to facilitate that. So as you see here, embryos, like I said, when they're bigger, they grow, when they arrive into the uterus, they grow exponentially. So around day 6.5, they're about 130 micrometers, so 700 micrometers. But then they double in size the next day already, and they keep on going like that. So the moment they arrive in the uterus, they grow because they have all these nutrients from the uterus, and they have all the space there, whereas once, when they're still in the oviduct, they keep having the same size as an oocyte. So this is a bit how embryos grow, and the first stage, we call them a morula stage. So that's how they arrive into the uterus. And then they start developing into what we call a blastocyst. And the reason is, if on the left, this is what we call a morula, and it looks a bit like a raspberry, and there's all those little balls are little cells. And you need to imagine that the embryo is fed by what we call uterine milk, and that is produced by the endometrium. So the uterus will produce food for the embryo. But it needs to go all the way into that little embryo. So it goes in, and those cells on the outside will eat, and then the next cells will have food. But if the embryo keeps growing like that, you can imagine that by the time the outside cells have had their food, there won't be much less left for the center cells, and they would die. So nature did something, and they did what we call blastulation. And basically what it is that the center of the uterus, if we go all the way to the right, is now filled up with fluid, and then it pushes all the cells to the outside. And like that, there's a lot more surface of your embryo that will be in contact with your uterus. So it's just helping your embryo to survive and get enough nutrients. And if you see here, this little blob of cells will become actually your embryo. And then this single layer here around will be part of your placenta. So even at day 8, there is already some cells that will become the embryo, and some cells that then actually will contribute to your placenta. So very early, you'll get already differentiation of your cells. So this is under the microscope. So this is that morula, and that is how they look under the microscope. And this is what we call then a blastocyst. So here you have that bit will become the actual embryo, and then this will become the placenta later on. So the next step is the actual embryo flush. And as I said, we do this around day 7 or day 8 after ovulation. So for example, day 7 would be if I inseminated the mare on Monday, we would do the flush the next week, Monday as well. Or if that would be day 7, or day 8 would then be on a Tuesday. And those on here are actually when I had a double ovulation and we were lucky to retrieve 2 embryos. So how do we do it? This is the uterus. We have here what we call the uterine body. We have 2 horns with 2 ovaries here. And this is the vagina, and here you have the cervix, all this. So this is the entrance to the uterus. And when the mare after ovulation, so this will be quite close to what we do, is we bring this catheter in all the way through the cervix, and then once you're through, at the end there is a little balloon, a little cuff. And so once we're into the uterus, we will inflate that cuff and we will pull it back against the cervix so it's nicely sealed. And so when we then we will fill the whole uterus with fluids, because the embryo is somewhere free in here. It can be here or there, we don't know in advance. So we make sure that it's nicely sealed, and once it's nicely sealed, we will put fluids into the uterus and the embryo will float in those fluids. So this is how it looks. We will have here again that catheter with the cervix here, and then here we have the little balloon. And so we start with fluids, and there's a clamp here which are both closed at the beginning. And so here you have a filter, which looks like this. And on the bottom, there is a filter. And so that filter will be smaller than the embryo. It's about 75 micrometer. So what happens, the fluids will go through, and on the filter on the bottom, the embryo will be stopped. So the first step would be filling the uterus. So we open the clamp here at the fluids, and the fluids will run into the uterus. And we will go rectally kind of to make sure that both horns are filled, because we don't know if the embryo would be in the left horn, in the right horn, or in the body. And once we're confident that the whole uterus has been filled, we close this clamp up here, and we open the clamp here towards the filter. And due to gravity, the fluids will go out of the uterus and will flow through this filter. And the embryo will be stopped by the filter on the bottom. And so once we're done, we will empty the contents of that filter into a Petri dish, and we will look under the microscope. And if we're lucky, we will find an embryo. And this was one I've done last year who had a double ovulation. And so we had 2 embryos. And if you look very closely here, this in that top little circle, you can actually see it with the naked eye, and sometimes you can actually see them already in the fluids. So while you're flushing, you're already getting excited because you know you did a great job at your insemination. And then the next step would be an embryo transfer. And what we first do is we kind of wash that embryo. And what I mean is we pass it through different droplets. And you can see that here in this dish, we will pass this embryo going from here and going through all those droplets. And the reason for that is that you want to make sure that there's no cells of your donor mare going into the uterus of your recipient mare and giving her a reaction there. And so what we do is we actually load the embryo in a straw. And so this is, imagine, well, there's on here on the top, those are the straws we use. And so what we do is we fill it up with an embryo holding medium here, and we have 2 little columns of air there, and in the middle we'll have fluid with the embryo. And the reason we use those air columns is just to make sure that the embryo sticks there. So if we kind of accidentally would flick it, it wouldn't come out. It just nicely fixed here. And then once what we do to transfer it is we use this stylet, and we go through the cervix, and we will push. Once we're sure that we're into the uterus, we will use a stylet, and this will push the cotton plug all the way up. and then the fluids will come and it will depose the embryo into the uterus. So how do we actually trick the recipients, her uterus, and thinking like, I'm actually pregnant, because we never inseminated her, and it's quite fascinating that we actually can think like, this mare is actually like never been inseminated, and now I have an embryo, let's carry this and pretend it is mine. The reason why we can do that is that actually the hormone levels of mares that ovulate and have been inseminated are the same as mares that ovulated and haven't been inseminated. So when a mare is in season, they're under influence of estrogen, and that will have the estrous behavior and everything. And once they ovulate, they will produce progesterone. So here at day 0, at day ovulation, they will start slowly producing progesterone. And around day 6.5, the embryo will arrive into the uterus. And up till here, actually, if the mare is pregnant or not, it's the same. But then the embryo, when it arrives into the uterus, it starts traveling around into the uterus. It will be in the left horn, it will go to the right horn, and giving a signal to that uterus saying, hello, I'm here, please continue producing progesterone, the pregnancy hormone. If the embryo doesn't go in here, or we know that if the embryo doesn't travel around, then a hormone called prostaglandin will be released by the uterus and will bring the mare back in season. And that's the signal saying, look, you're not pregnant. You need to come back in season, because if you don't come back in season, you won't have a chance to become pregnant. So it's the embryo that gives the signal to be pregnant and to continue producing progesterone. And that's why we need to have this synchronization of the donor mare and the recipient mare, so that the embryo arrives at the right time into the uterus of the recipient mare and can give the signal to the recipient mare, please continue producing progesterone, because I'm here and I would like to become a foal one day. And then the final step, if we're successful, is actually a pregnancy diagnosis. And in general, we do a pregnancy diagnosis on day 14, but because we did the flush at day 7, we do roughly the pregnancy diagnosis a week after the embryo transfer was done. Although I must admit, I don't have so much patience. I'm waiting a week, so I often do them at 5 days after, or at day 11, and just have a little peek if the embryo is there already. Because after the hard work, you kind of want to know if you were successful. So what to expect? So this is the pregnancy rate per cycle, and basically about 50 to 65% of the embryo flushes result into an embryo. And of those, between 70 and 90% will be pregnant after transfer, resulting in about 35 to 59% of positive pregnancies. Last year we did over 100 flushes, and we had about, I think, 56% positive flushes with an 83% pregnancy rate. So just to give you an idea, this, like I say, you need to start with having an embryo to be able to do a transfer. The success rates depend a bit on what kind of mares you're using. If you use a lot of older mares, subfertile mares, that percentage might be lower. If you use a lot of young mares or mares without any issues, that percentage might be higher. So I had a mare that we did 5 flushes on 5 embryos. And then you have the frustrating ones where you do 4 flushes and have 1 embryo. And the more you do, the more you get like an accurate number there. And then the transfer is very veterinary, like experience dependent, and it's all, a lot of things like that are in the details, in working very clean, in managing that embryo very nicely, managing the cycle of your donor mare, choosing the right recipient mares, making sure that that recipient mare has a perfect, nice, clean uterus, because that embryo needs to develop there for about 11 months and it doesn't want to arrive in a dirty bedroom for 11 months. It needs to be clean, and you just want to make sure that everything you do is ideal for that little embryo, and to really optimize your chances there. So why would we choose for embryo transfer? Why would you go through all that hassle of embryo transfer? Very popular in competition mares. Historically, we always looked at the stallions. The stallions' genetics seemed to be more important, and the reason was mainly because mares could only, when competing mares weren't able to have offspring. But now with embryo transfer, and I will shortly speak about ovum pickup, we have found a way of producing multiple offspring of competing mares or retired mares as well. But more and more people have found that mare genetics are at least as important as the male genetics. So competing mares are a big group of doing embryo transfer. Another reason, young mares. I sometimes do embryo transfers on 2-year-olds, where you kind of want them to grow a bit more, to develop that pelvis a bit more, that abdomen, just to make sure that they have enough space. But you could actually do an embryo transfer on those. Another thing is nursing mares that fall late in the season, because every time you kind of move around a bit, and if you then do an embryo transfer on one of those mares, she will still have offspring, but then the next year you can start early in the season and you win a year. So you can actually start in March instead of if she only falls in August, and then by the time she's back in fall and everything, it might not be ideal, but then you can do embryo transfer on one of those mares. And then mares that are unable to carry a pregnancy or give birth to a foal for certain different reasons. A used uterus like endometrial cyst, although we can laser those cysts, but sometimes they're as well a reflection of a uterus with other issues. Like this is a picture, those or those glands that I was talking about that will feed the uterus and the embryo, and if they don't produce enough what we call uterine milk, then they won't feed that embryo nicely, and that embryo won't develop properly. Another reason might be a mare that had a pelvic fracture, and so the birth canal is too narrow to give birth, or a cervical laceration of a previous pregnancy, and those mares might still get in fall. But if the cervix is not sealing really nicely, and then the fetus develops and getting more heavy and heavy, it starts pushing against that cervix, and often it just later during pregnancy that they will lose it, and those are candidates for embryo transfer. Subfertile mares can be very frustrating, because we need to keep in mind that the embryo still will be for a certain period of time in the uterus. So if you have a uterine infection, that can infect as well the embryo, or it's possible that there's other reasons why the mare doesn't produce an embryo. So those can be frustrating and can be costly as well by repeated cycles of insemination as well as flushes. And then another reason might be that you want to obtain several foals in the same year, and that is possible with embryo transfer, as I said earlier. And then it might be that you want to obtain embryos for freezing, although I would say that you can do it. It's just a lot of work involved, and if you really want to have frozen embryos, I kind of at the moment would rather advise to do ovum pickup and have multiple embryos like that, because it's a bit more efficient to do it like that. Another thing would be fetal sexing, so or actually embryo, not fetal, actually embryo sexing, where like I said, that embryos, those blastocysts have fluid, and we can aspirate that fluid and then determine the sex of that embryo. But that's not something we routinely do. Certain diseases can be determined on embryos as well. So as they do in human medicine, they can be done on embryos before you transfer them. So yeah. So what is embryo freezing actually? Embryo freezing, we're going to talk about two different embryos. We have what we call the morulus here, and then we have those blastocysts. And as you see, as I said, this is actually a fluid-filled cavity, and they're so much bigger as well. And the problem with those fluid-filled things is a bit like putting a Coke in the freezer and thinking like, oh, well, I'm just going to go and do this and that and forget about it. And then they look like that. And that is exactly what would happen if you would just freeze this, because ice crystals, even though it is just a water molecule, is bigger than a fluid water molecule. And it will just expand, and it will have all these little crystally points, and it will break, it will actually explode your embryo. And the only way we can actually freeze those embryos successfully is by aspirating all that fluid out. And this is an embryo I've done a few years ago, where I aspirated the fluid, and then you aspirate all that fluid out so it can't form those ice crystals. And then we use cryoprotectants, so kind of antifreeze, to protect the cells, and then freeze them like that. But it's not something we can routinely do. We need a special microscope, which we call a micromanipulator, where we can actually aspirate this fluid successfully. The embryos we can freeze or what we call the morulus, the smaller ones. So when they're smaller than 250, we can commercially freeze. We're using a commercial embryo vitrification kit, and we can do that. And we have about although you can do it, your pregnancy rate after transfer of frozen embryos are about 15% lower than when we do a fresh embryo transfer of in vivo produced embryos. And I'll later talk about the OPU embryos that are frozen, and those success are actually the same as when we would do a fresh transfer. And the problem is, as I showed in that earlier slide, is the moment when the embryo arrives into the uterus, it starts to grow. And it's quite variable when that embryo exactly arrives into the uterus. So there is a chance that you plan to freeze an embryo, and the embryo, you do a flush, and the embryo is too big. And then you're there with that embryo, and you need to do something with it. And so it's quite tricky to do. I can do it, we do it sometimes. Although since we're doing ovum pickup as well, if people really want to do embryo freezing, we advise to do ovum pickup. So we're doing less and less of the actual embryo freezing like this. So I'm just shortly going to talk about ovum pickup as an alternative for embryo transfer. Because I think talking about ovum pickup would be a whole other talk, and I could probably fill in a complete evening about it. And I think we will do that in the future as well, and do another webinar solely on OPU and ICSI. But I do think that at the moment it would be good to give you a little introduction on OPU, and hopefully get you triggered to come and watch the next webinar on ovum pickup we will give in the future. So what is ovum pickup? So the first thing would be the oocyte collection, and it's what you see here. And so basically what we do is we kind of aspirate all the little follicles which contain one egg out of the ovary. And then those eggs are then transported to a lab, and they need to mature. And what we mean at oocyte maturation is actually we will mimic what happens into a growing follicle. So we will expose the oocyte to the hormones that are normally naturally in the mare during her season. And then those oocytes that are mature, that are ready to be fertilized, will be fertilized by a special technique. And this technique is called ICSI. And what we mean with ICSI is intracytoplasmic sperm injection. And we actually inject one little single sperm cell in each egg. And this allows us to use just a tiny bit of semen, just not even a whole straw of frozen semen, but even a tenth of the frozen semen, a tenth of a straw. We can do multiple oocytes because we will inject one single sperm per oocyte. And sometimes we have about 30 oocytes per ovum pickup session, and we would have different mares. And so even the oocytes of different mares can be done with a little piece of frozen semen. So it's a very efficient way of using stallions that have deceased, or there's a limited amount of frozen semen available. That is a good technique to use. And then the embryos are, so the next step would be those oocytes that are fertilized need to culture. So they go in a special dish, in an embryo culture, a special medium, in an incubator. And what I said, the first development in vivo produced embryos. So when we normally inseminate a mare, it will develop the first 6.5 days, as I said, in the oviduct before it enters the uterus. We will mimic this in the lab. So the embryo will grow in a petri dish. And then once they are an embryo, we will freeze them. And because they're a bit smaller and they're different, they have less water, they're smaller, they freeze very well. So we know that if we do a fresh embryo transfer of those ICSI embryos, so of those lab-produced embryos, or we do a frozen embryo transfer, they're having the same pregnancy results. So so that's why I say, if you want to freeze embryos, go for ovum pickup and ICSI, because you will have the same results. And then the embryos are stored in liquid nitrogen, such in the same way as we would store frozen semen. And we can thaw them, and we only thaw them when our recipient mare, we're 100% happy with her. If we're not happy, we leave the embryo in the tank, and we say, look, come back with a different recipient mare. There is no rush. It's there, it's safe, and that's how we do it. So why would you choose actually for ovum pickup and not for embryo transfer? Mares with certain subfertility issues, and mainly what it is that because almost every step is done in the lab, you circumvent the whole reproductive system. The embryo, the oocyte doesn't need to be released in the oviduct. So if you have issues in the oviduct, that won't be a problem. The oocyte is matured in the lab. So if you have hormonal problems in the ovary, that will be overcome by culturing those oocytes in the lab. If you have a uterine problem, it will be overcome because you're doing it in the lab. As I said, frozen semen, it's even more than 1/5. You can use a really tiny bit of a single straw of frozen semen per ovum pickup session, and even for multiple mares on the same day. And then stallions with reduced fertility, because ICSI is actually a technique developed for male infertility. So from oocytes, from sperm cells that have problems penetrating the oocyte. And in contrast with embryo transfer, where the donor mare needs to be in season, so it's a seasonal thing, OPU can be performed year-round. So there is no season to do ovum pickup, although at the moment we're doing less during the summer just because it's so busy with the actual breeding work that it makes it very difficult to combine, but it can be done year-round. And then another thing is that with ovum pickup, there's no hormonal manipulation. So not like women that undergo IVF cycles and they need multiple injections. Because we do all the maturation of those oocytes in the lab, there's no hormonal manipulation. So it might be good for certain mares as well that have issues with coping, of coming in season constantly, the mares that that have problems with that, because when we do an embryo flush, the mare has been inseminated, so she was in season. Then we would do a flush 7 days later, and after the flush we would give her a prostaglandin injection to bring her back in season, so she would come back in season again. And so some mares have a bit of issues in coping with all those hormones, so those might be good candidates. And like then as well, the frozen embryos can be transferred as desired, and then there's little interruption as well with the competition. With the ovum pickup, we can really plan it. It's not like, well, the mare is in season now, we inseminate her, and then next week we need to flush her. And it has happened to me, and it has happened to some of my colleagues here as well, that you inseminate a mare, and actually a few years ago, before I was here, I had a mare inseminated, and then they decided to go to a show. So I ended up on the show ground in the stable, flushing the embryo out there. So that's less than ideal, whereas ovum pickup can really be planned. And then finally, to conclude, what is the chance to have a day 45 pregnancy? This is a study done by my old colleagues back in Utrecht, where they compared natural mating, artificial insemination, embryo flush and embryo transfer of ovum pickup, ICSI, and embryo transfer. Sorry. And their natural mating had the highest results, and then we saw that AI and embryo flushing is roughly the same, and then ovum pickup was at 37, a bit lower. However, with ovum pickup, you can have multiple embryos, and we have mares that had 12 embryos last year. So by having those multiple embryos, you can even go above this, just because if you would transfer all those embryos and those transfers were successful, then your ovum pickup might be a good choice. But I do think making the choice for AI or embryo flush or OPU, it's really a mare dependent thing. If you have a mare that constantly produces an embryo for embryo flushing, stick to the embryo flushing, it will be cheaper. However, if you have a mare that needs multiple cycles to produce that one embryo, it might have been more cost efficient to go for ovum pickup and ICSI, and go for that. And it all depends on the individual basis, and that's why we always have more than happy to have discussions with our clients and see how we can do it and how we can optimize the chances and make sure that at the end of the whole procedure, you hopefully end up with a gorgeous little foal. And so yeah, that's my talk of this evening, and I hope you enjoyed it and you learned something, and I'm sure that Simon will have plenty of questions for me.
Kaatje, amazing job. First thing, can you share me back onto the screen?
I can do that. Let's see. There you go.
You stay on the video, hopefully.
No, you need to enable me.
Can you not keep your camera on? I'm on, but now I can't see Kaatje because it was a fantastic talk. And I have so many questions for you for the next 45 minutes to an hour that I really would love to see you as well.
You need to enable me.
We'll just try to get Kaatje on the video as well, and then we'll start going through all those questions. Can you come on as well, Kaatje?
You need to enable me to, as a host, you need to accept.
I think I have. I'm very sorry for these Technical difficulties, perfect.
I'm
Now we're all there. You were there and
I'm
disappeared
back,
again.
yeah.
There we are. Luckily, we're better vets than technically gifted people. So, Kaatje, there is so many questions about so many topics, and I think I would like to start really with the basics. So one question was, can you tell us one more time, what is a fetus and what is an embryo?
So an embryo is an earlier form. So you have, so once the embryo the embryo will develop, and I think it's about 40 days. You start talking about a fetus. It's just kind of so it's just kind of a developmental stage. You start with an oocyte, then the oocyte becomes an egg when it's fertilized, and then it starts. We call it a zygote once it's kind of starting dividing, and then it becomes an embryo. And an early embryo is called a morula, and it becomes a blastocyst, and it becomes a fetus, and then hopefully it becomes a foal, and that's how it goes. Yeah.
Very good. So I hopefully we cleared that up. And then another question is, so the genetic material in embryo, is the genetic material in an embryo influenced by the recipient?
So the genetics come from the donor mare. So your donor mare is so the embryo, the genetics of the embryo are from your donor mare and your stallion. However, it is very important. You have the genes there, but certain little twitches are switched on by the nutrients into the uterus. So you need to have a healthy uterus in order to kind of feed that embryo properly. So all those little tweaks, and the development of that embryo happens perfectly. So that's why I also think that sometimes you get a recipient mare and you think, oh my God, that looks like, you kind of want to have a healthy recipient mare, and you need to feed that recipient mare as it is the donor mare. You want to make sure that it gets this really good nutrition, that the right minerals, the right everything, so it develops nicely, because you give the genetics of your donor mare, but it's your recipient mare that needs to cook it properly. So
that's right. So you would argue, like a lot of breeders do, that ideally you want a good quality recipient that is kind of doing the similar thing like the mare giving the embryo. Can you specify one more time, because there are several questions, what is your ideal recipient mare?
I do use maiden mares, but sometimes I do like, if I have the choice and people to use a mare that had one embryo or one foal before, because then you know a bit the temperament of the mare, and size wise, I don't do hands very well, so about 170, 160, 170 mare. I'm not too fussed on size. I'm more, because some smaller mares have a nice enough space. I think important when I scan the uterus, I would always scan the uterus. When people give me a recipient mare, I would scan the uterus, look if the whole aspect of the uterus is fine. If the ovaries look normal, and I would always do a vaginal exam as well, because I like to do them in actually in diastrus, so when they're not in season, so I can really nicely palpate that cervix as well, especially when they had a fall before. And it's nice when they're not in season and the cervix is closed, to kind of palpate that cervix to make sure there's no lesions there, and you can miss those lesions when they're fully in season and that cervix is very relaxed. And when they're not in season, the cervix is nicely closed, it gives you a lot of information. And so temperament is for me important, and a good healthy uterus.
What about the age of the recips?
So we know that fertility goes down around with older mares. So if you take a recipient mare, you would go for like a 4-year-old to a 10, 12-year-old. But if you have an older mare, if you wanted to repeat it, you kind of want to continue using them. But I have used older mares as well, of which I know that I've used them for 4 or 5 years in a row, and they constantly, every embryo you put in there, they're pregnant, and if falling went fine, I would be okay to use them as well. But if you say the ideal recipient mare is between kind of 4 to 10 years old and had one fall and all went fine, and that's what I would choose for. But in an ideal world, but we're not always living in an ideal world, but if you can choose, that is what you would choose for me. I'm not sure if you agree, but
yes, of course I agree. you are the main speaker. If I really don't agree, I will say. Moving on now, kind of to the donor mare, there's a lot of questions, you know, and I think it's important to reiterate it again. So can we use frozen, chilled, or fresh semen natural covering to produce embryos in donor mares?
Yeah, I really don't care what kind of semen as long as it's fertile semen, and that's what I said, that for me the most important is your management. You want to make sure that the embryo, that your cycle is properly managed, so you inseminate at the right time, or you live cover her at the right time. And to be fair, I think 80% of what I do, or even more, is frozen semen. And look, I've done last week, the 2 flushes I've done this year are frozen semen with 1 straw, and we have embryos. And I think with frozen semen, it's just important that you do right management as well. And what I often say as well, I rather use good quality frozen semen than bad chilled semen. And a lot of people say, oh, chilled semen is better, and I don't always agree on that. With the benefit with frozen semen, and especially with stallions that you've used before, is that you know kind of the quality you're using. You need a bit more, there's a bit more work for your veterinarian on doing that. The vet needs to scan a bit more to do the actual correct timing as well. We put the semen with frozen semen, we do what we call deep intrauterine insemination. So we make sure that we know at which site the oocyte, the mare will ovulate, and then we will depose that semen really into the horn next to the entrance of the oviduct, so it's ready to go. And all those things enhance the chances of having a successful embryo flush. But I have
a question to the timing. So when you breed with fresh semen or natural covering versus frozen semen, does this affect the timing when you flush the mare?
It's basically what it is. It's not that people often say that frozen embryos are smaller than embryos produced by chilled semen. But what it is exactly, it's because we're frozen semen, we often inseminate after ovulation. So semen, when we bring it into the uterus, it needs to undergo certain changes before it can fertilize. It needs to. And so when we have chilled semen, we bring it into the uterus, and it will travel towards the oviduct, and it will sit there at the entrance of the oviduct, and it will be released in waves. And then once the oocyte arrives into the oviduct, once at ovulation, it's ready there to fertilize. So fertilization will occur quite fast. With frozen semen, we often inseminate after ovulation. So the oocyte ovulates, enters into the oviduct, and then we bring the semen in. So the semen still needs to kind of swim to the oviduct, undergoes those changes in order to be able to fertilize, and then fertilize the oocyte. So that's why the embryo is what we call smaller. But if we would do the same with chilled semen, so we inseminate after ovulation, the embryo will be smaller as well. So with frozen semen, because we often do it after ovulation, I would do probably day 8 my flush. So day 8 after ovulation. So if I inseminated the mare on a Monday, I would do the flush on a Tuesday. If I detect my ovulation with chilled semen, so with chilled semen, often if I order my semen I scan my mare on Monday, I will order semen, I'll give her her ovulation-inducing agent, I inseminate on a Tuesday, she ovulates on a Wednesday, so I would flush next week Wednesday, if that makes sense.
And if it was frozen, you would do it on the Thursday, basically.
Yeah, so you calculate on the day of insemination, but on the day of ovulation, so chilled semen day 7 or day 8, if that, like, I don't really...
Does the age of the donor affect when you flush? Would you, a younger mare, flush early? And also maybe one more time, is there a difference in quality of embryos we get from young donor mares versus older donor mares? First, the timing of the flushing, that's the first part of the question.
So older mares tend to have a little bit of smaller embryos, so you would flush it. I would often flush a day later, so day 8 or 8.5 with frozen semen. And then indeed the age of the donor mare influences the quality of the embryo and mainly the quality of the oocyte actually. We know that older mares have higher chances of embryonic loss. I've spent 4 years of my PhD on maternal age. So basically, just like women that are, we know in women after the age of 35, they have an increase of what we call aneuploidy, and a very known aneuploidy is trisomy 21 or Down syndrome, where you have a chromosome too many of chromosome 21. And we also see that in horses, and that starts at the age of 15 roughly, that the fertility decreases. And so we have a higher embryonic loss of embryos of older mares. However, interesting, if we have embryos from ovum pickup, so the embryos of ovum pickup of older mares are of the same quality as embryos of younger mares, which is quite interesting, I think.
Oh, that's very fascinating, that fact, you know, I mean, and that also is such a promoter, you know, for in vitro fertilization in older mare, that actually the quality of the embryos doesn't suffer versus, you know, when we do embryo transfer normally. Moving on a little bit to the synchronization now of the recipient, you know, so can you one more time reiterate, you know, what's the ideal window? So we prepare now our mare, we know we're going to flush either on day 7 or day 8. So what synchronization do we want to achieve? What's your ideal synchronization kind of?
So what I normally do is when the moment I detect ovulation in my donor mare, I will give an ovulation-inducing agent to my recipient mare. And so that means that if we're going for Monday again, Monday is day 0 in my donor mare. If I give an ovulation-inducing agent on that Monday to my recipient mare, she will be ovulate 2 days after my donor mare. That's what I normally do. And I kind of rather have them ovulating after my the recipient mare ovulating after my donor mare. And the thing, for example, if I have twins, so we have a double ovulation, and I have a bigger one and a smaller one. The smaller one I will put in a recipient mare that ovulated a shorter period of time ago. So maybe the one, if I have a mare that, if I have twins and I have
of different sizes,
you're talking of different sizes.
Yes.
So I have a bigger and a smaller embryo. The smaller embryo I would put in the mare that ovulated the shortest period, because if I have a mare that ovulated 5 days ago and 7 days ago, I will put the bigger embryo in the recipient mare that ovulated 7 days ago, and the smaller embryo in the mare that ovulated 5 days ago. So just kind of try to kind of synchronize them as well on developmental stage of those embryos, because those twins are on what we call, they're not completely synchronized. So you can have one ovulation and then the other ovulation happens a day later. So that small embryo can be a day a day younger.
A question that has come in is, how do you treat the recipient, that is getting the embryo? Do you give those recipients any treatment at the time of the transfer?
I don't give them anything. The best
You don't sedate them?
Oh yeah, sorry. Yeah, I do sedate them because the mare is what it called diastrus. So, and the natural reflex of a mare in diastrus is to kick to the stallion. So there's not really supposed to kind of manipulate the cervix or anything. So I sedate them very well. And what I do, I'm very, very particular in the cleaning of the uterus, of the vulva. So I make sure there is no soap entering into the vagina, and that it's very, very, very clean. Because when a mare is in season, her immunity of her uterus is very high. She's under influence of what we call estrogen, and she's expecting to have semen arriving together with bacteria and other things. The cervix is open, is an open connection kind of with the outside world, and so the immunity of that uterus is very high. And because
sorry, what soap do you use? Just really some practical questions when you say you want to really clean this recipient very well.
I use a very diluted iodine scrub. So I kind of put 10 mils in half a liter, and I make sure it's very well rinsed. And then I have something I started using at university is called Sempercept. It's kind of a very diluted, specially developed concentration of chlorhexidine, but very diluted with alcohol as well, just to wipe it off and to make sure that it's all very clean. And you don't want, especially not have any soap or water in contact with your embryo, because it will just kind of kill them. But like I said, so the uterus, when there is in season, the immunity is very high. And then when she ovulates, she goes under influence of progesterone, and the immunity is very low. And so it cleans up the uterus under influence of estrogen. And then once it's all clean, it's under influence of progesterone. So if you bring something into the uterus, then any contamination, it will release prostaglandin. And so your mare would lose the pregnancy. So I think the most important thing is clean. It's just working very, very clean. Make sure that your embryo is clean, that your vulva is clean, that everything you do is very clean. And people that work with me think that I'm very a bit OCD there, but I think once you've taken that embryo out of the uterus, it's of the donor mare, it's our responsibility as a vet to take very good care of it and put it in a uterus and take very good care of that little embryo. It shouldn't be out of the uterus, just to take care of it and make sure that it's
I can't agree more, Kaatje. Definitely. I mean, there's maybe one thing I do different, but everybody has their own techniques. I tend to treat my recips with some flunixin before the transfer as well, exactly because what Kaatje was saying, that actually just the catheter going through the cervix, does release some prostaglandin, and that theoretically could cause luteolysis. So actually giving flunixin when the mare is scrubbed up, I think, is not wrong, with all the efforts. But otherwise, I would definitely agree. There also have been now some questions about the flushing process. So what is actually the medium you're using to flush the donor? And what's the temperature of that fluid?
So we have the fluids at body temperature. So kind of, you don't want to have cold fluids going in there, because if you use very cold fluids, you get what you call shock and have like a little shrinking embryo. That's just like, oh my, that's not what I really like. So we tend to do them at body temperature. You can use either commercial embryo flushing medium or actually lactated ringer or Hartmann's. are absolutely fine to use to do embryo flushing with, the ideal thing is just osmolarity, so it kind of need to be buffered with lactate, so lactated ringer, and that is absolutely fine. And then once the embryo is out, we transfer the embryo in a holding medium, which is a very simple medium, which is just buffered, and the function of it is just to kind of that holding medium is basically first in the washing, removing all the cells of the uterus that might be attached to that little embryo, and as well keeping it quiet, kind of just keeping it happy in its until you're going to transfer it. And then while you have it in your little dish, it's important that you keep the temperature okay, so it's not cold there, so you kind of want to keep it at a correct temperature. So the room, I don't mind to have it at room temperature, but not lower, and I always protect them from the light as well. So you don't want to constantly have light going on there. So if once I'm done and I found the embryo and I'm ready with washing the embryo, I will cover it, because you need to think that little embryo was supposed to be in the dark, normally only should come out of there 11 months later, and now you suddenly take it out, so cover it for the light and make sure it doesn't.
And how long can that embryo be outside the donor mare, in that holding medium, as you call it?
So we transfer for a day, so you can keep them out there for a day. But if you take them longer than a few hours, you will slowly cool them down. So it's important to cool them down then, and make sure that they're transported correctly, so you don't heat them up. So we use special transport boxes, either an Equine or a clipper box, that are specially designed to maintain that temperature, to slowly cool that embryo and protect it from the outside temperatures.
Yeah, very good. That would be for the flushing procedures. So if you have any more questions, please send them through. At the moment, there's about 70 questions outstanding, so brace yourself, Kaatje. But I just try to go through the procedure as we go along, kind of, and then maybe go back. So now I would like, some questions in your talk were about vitrification of embryos. And can you explain one more time why we could really only vitrify early blastocysts or marula versus expanded blastocysts, the bigger ones?
So with the commercial ones, with the commercial kits that are available, so without using special equipment, we can only do the small ones. And the reason is that they are smaller. So if you have something small and you put it in the freezer, it will freeze faster. If you have something big, it will take longer time. And the faster they freeze, the less ice crystals they form. The other thing is that the bigger the embryo, the more fluid is in the embryo. And the fluid, as I said, fluid will form ice crystals. And ice crystal formation, if you see a little crystal, it has all pointy points, and it's bigger than a water molecule, so it kind of bursts the embryo. So those are problems. So we can freeze large embryos, but then we need very special, the same kind of microscope as we use for the ICSI, where we really aspirate that fluid out, which makes then a smaller embryo with less fluid, and those we can freeze.
And can you run us through, for people who haven't done freezing, just very simply, how does actually vitrification work? So there we have our little morula in a dish. So what's going to happen till it ends up in the liquid nitrogen tank?
So we use cryoprotectants. And what is a cryoprotectant? Is something that it's an antifreeze basically. It will avoid those ice crystal formation, because every cell has some fluid in there. So if you have one cell, you think of it, cells like what we call homeostasis. They want to have the same concentration of things outside and inside. So when an embryo is, or we're just going to talk about one cell freezing and make it maybe a bit easier. If you have one cell and there is just water inside and water outside, and now we add that little cell in what we call cryoprotectant. So what will happen is water will flow out and cryoprotectant will go in, and it will start to have, and it will happen until it has the same concentration of cryoprotectant inside and outside of the cell. So that's the first thing we do. If it's not clear, just stop.
Yeah, no, very good, very good.
So the cryoprotectants will go in and the water will go out. So the cryoprotectants take in the place of the fluid, of the water, just to make sure, and cryoprotectants won't form those crystals. So it will protect the cell of being punctured by ice crystals. And then once that has happened, you can even remove even more by non-permeating cryoprotectants, but then I go a bit too much into detail. But
it's just in the big lines, you know,
then,
that
so
we can understand what happens.
Once that has happened, once those cryoprotectants are in there, and you have those cryoprotectants in there, you kind of put it in the liquid nitrogen, and then it will cool down very fast.
And how does the thawing happen? Is that like frozen semen that we just put it in a water bath for 25 seconds and it's all good to go?
So with the commercial kit for those commercial embryos, they have the medium ready to do that. For example, the ICSI embryos, it's different steps. We go from a high concentration of this, so the concentration of the cryoprotectants in that embryo is high. If you would put it in directly in holding medium again, the permeability from the membrane, so from the outside of the embryo for water is higher than for cryoprotectants. So what do I mean with that? Is that cryoprotectants flow out slower than water flows in. And so what will happen first is that if you would put it directly in holding medium, the holding medium will go in, but the cryoprotectants doesn't have the same speed, so it will go out slower. So what will happen if you would do that is that it just bursts, because the water goes in, it swells and it bursts. So what we do is we slowly put it in from a higher concentration, and we'll slowly kind of let that effuse out of there and just kind of effuse that cryoprotectants out without bursting the cells. And that's how we do it in multiple steps.
Yes,
just kind of, yeah.
That's kind of where I wanted to get to. It's not just quickly going into the water baths. It is a 20-minute gradual process with several media, because we basically need to get back to an embryo at 37 degrees. Yes, thank you very much. I think that explains it. One more question to freezing embryo. So if I don't have a recipient and I need to freeze my embryo from the donor mare, because you want a slightly smaller embryo, would you maybe flush that mare slightly earlier?
So what I like to do for my, if I do frozen embryos, is actually, even with chilled semen, inseminate post-ovulation, because then I have a bit more of an idea when fertilization actually happens. And I would scan the mare every 6 hours, or even more often. And then I would calculate from the day, or from the moment of ovulation, and I do them at roughly, I actually do them almost at day 6.75. So really that moment. And just because if you're too early, the embryo isn't there, and you could say, okay, I flush again, and I've done that. But if you flush an embryo and you've done a flush a few hours ago, you will have some reaction. It's just a sterile inflammation of your uterus, which you also get if you scope. That uterus will react to it, because it wasn't supposed to kind of be distended like that. So if you do a flush then, it's kind of not really nice to do. And if you do it too late, you won't have an embryo. So you kind of need to find that way. And I do warn the clients that when we freeze embryos, there is a chance the embryo is too big. It's just the moment it arrives, it's in this rich environment with all this histotroph, so the uterine milk present, and that embryo will just thrive and suddenly has the space because it was in this oviduct and it didn't have space to kind of grow, and suddenly it has the space to grow. So once it's there, it doubles in size very quickly. And so very close monitoring, even closer than when I do a normal flush. I would monitor that donor mare very, very closely.
I think that answers that question really, really nicely. Now, moving on to the new exciting technique of ICSI embryos. The first question was, can you tell us one more time, actually just to start it a little bit simpler actually, when we aspirate the oocytes, how many oocytes do we expect to aspirate from a mare, and what's kind of the ideal time of the year, you know, for those mares to come into a clinic to do oocyte aspirations?
So the first thing we do is scan the mare and look how many follicles there are present. And we know in mares that are transitional, they have, because they don't have what we call the peak, the LHP. When they transition, now what I mean with transitional, there's two periods. There's spring transition and autumn transition. So during spring transition, in the winter, most mares stop cycling and they go in what we call an estrus, and the ovaries can become tiny olives and just kind of have a little hibernation. And then the transition of going from not being cycling at all to their regular cycles during spring, in that period when all the hormones starting again, that period there's a lot of follicles present, and those are periods that are very ideal. And we have about 60-70% recovery rate of those oocytes. And what I mean with recovery rate is that we aspirate the fluid out of the follicle, but we can't see the actual oocyte. So we aspirate the fluid, and the oocyte is tightly attached to the follicular wall. So we, under ultrasound guidance, we insert the needle into the follicle, and then we will move the needle just because like that we can scrape the follicular wall and hopefully like that detach the oocyte. Women that have undergone.
Sorry, interrupting you. So for breeders at home, you know, what number of follicles would you like to have in a mare to bring her into a collection center?
I would say at least 15, 10. But I mean, some mares don't produce 15. We have mares at.
Can we maybe do one more time the math? So let's say I bring my mare in, she has, to make it really simple, 20 follicles on there. So what are now the numbers, you know? So we aspirate 20 follicles.
You would roughly have 12
oocytes, and then what, but they still need to be matured, is that correct?
So yeah, so it depends a bit on the mare, but you would have, if you have 20, if you have 15 oocytes, you would have roughly 1 or 2 embryos, if that makes sense. So you go from your oocytes, and then you have 60-70% will be recovered, and then about, depending on a bit of mare, more than half will be ready to be fertilized, and then they start cleaving, and then they go to an embryo. But we have mares that's statistics. So ideally you have to have a proper chance 15 or more oocytes. If
I have a question here from a breeder who says we had 10 follicles, 10 oocytes shipped to the maturation center, and none of them developed into a fertile embryo. So what's going wrong in those mares?
So there's a few things. There's a few things to think of. One thing is, and that's a study done in uterus as well, is they looked at what factors are important. One thing is the mare, and we know that the results are often repeatable. So if I have a mare that produced embryos, she's very likely to produce next time with ovum pickup again embryos. If, however, she didn't, there's still a chance that I would repeat the procedure and go again. However, I would use in those mares a stallion that has been proven to be okay with ICSI. Some stallions we know have better results with ICSI than others. And interestingly is those results depend on the center which doing the ICSI as well. So we know that certain stallions produce better results in a certain place than another for whatever. And the thing is with the ICSI, it's very precise. It's a lot of things happening in the lab, and it's all very, very, very tiny little details. And that's the same with the ovum pickup. It starts there. The ovum pickup, how it's done, is very important as well. And a lot of people think, oh, once the eggs are, it's only the part in the lab that is important, but it's as well, it starts with the moment you bring the mare in how you aspirate the oocytes, how those are manipulated, the temperature during your manipulation, how they are handled in the lab, how you expose them to temperature changes, how you wash those oocytes, how you hold the oocytes, how clean you work, how much you protect them from the light, how you pack them for their transport. Every little step in this long chain of things that I explained is important. So it's not just getting those eggs out and getting them in Italy or in another place where you do the flushing, but it's very important that every little tiny step will help with your successes.
And for ICSI to then fertilize those mature oocytes after 6-7 days, has the sperm to be frozen?
It doesn't need to be frozen. It can be chilled. However, often frozen semen is used. It's just but it can be used chilled as well.
And is it true that you can only use a small portion of a straw? So you can have one single straw and you can cut off that straw, yes, and use that straw several times and refreeze it? So
you don't refreeze, well, you can do refreezing, but basically what we do is if you have your straw, you just keep everything frozen and you just cut a little piece off and you tore only the little piece. And there's millions of sperm in there and you only need one for oocyte. So depending on the quality of the semen, you can use, with that little piece, even do 3 or 4 or 5 mares or more and fertilize all those oocytes. So for example, and a very known example for those ICSI is Choco Blue, where there's only a few straws left, and there's really what we call Choco Blue days, where centers here at Sussex Equine, but as well colleagues of mine in Utrecht and in Holland and in Belgium are doing ovum pickup on the same day. So we kind of communicate with each other as like, okay, are you doing ovum pickup this day? Yes. And that day you can join in and you buy, instead of buying a straw, you just buy an ICSI session. And so there's going to be one piece taught, and all the oocytes that come from different centers will be fertilized with that piece of straw.
Yeah, fascinating stuff, and unbelievable use of one single straw, you know, and no wonder the prices are going through the roof, you know, for these individual straws. And fascinating information. I have to say that certain straws only work in one center really well and not in another one. I mean, that is totally new to me as well, and I think very, very interesting. If it comes to transfer now of frozen ICSI embryos, what is the synchronization of the recip looking like for ICSI embryos? So when we transfer here at Sussex Equine Hospital frozen embryos for ICSI, how do our recips need to look like?
So I do them exactly 4 days after ovulation. Yeah.
Why is that? Why are they much closer to ovulation than in conventional embryo transfer?
They're just small. They're smaller, and they're just kind of an earlier state. They even though we culture the embryo for 7 to 10 days, they're just smaller. If you compare them in size, they're much smaller, and that's why they've done many studies where they compare the results with younger and so earlier ovulations and later ovulations and the pregnancy rates. So the pregnancy successes are the best if we do them 4 days after ovulation.
Very good, thank you. Next question, a bit of comparison question. Someone asked the difference to cattle embryos, you know, where we can flush a cow and get 20 embryos. Why can we not superovulate in horses?
So people that are like the vets that study equine reproduction, everything you know from cattle, I always say to my students when I was teaching, like, forget everything you know about cattle, because horses do everything different. And a good example is indeed this superovulation. Women, pigs, cows, they have their what we call the cortex that is on the outside, so where the follicles and the oocytes grow are on the outside, and the blood supply, which we call the medulla, where the blood and the nerve supplies and everything is on the center. So if you have a cattle ovary, we'll have all the follicles all around. If this is the ovary, they will be all around. And if one ovulates here, it will go to the oviduct. It will ovulate at the place where the follicle is. Horses, they thought, well, that's too easy. So they decided, let's put everything inside out. So the follicles grow, the cortex where the follicles grow are in the center, and then the rest is on the outside. And only one place, which we call the ovulation fossa, which is on here, up there, that is what we call the ovulation fossa, and only the follicle can only ovulate there. That's only the place where they can ovulate. So if we scan a mare, we often see just before ovulation, the shape will change. So actually that reason is that follicle will grow towards that ovulation fossa. And an ovulation occurs, so that ovulation is actually that bursting of that follicle will burst and the oocyte is released. And there is blood that will produce some blood as well. So the idea, probably it is, you have a blood clot there. And so then even if another follicle will ovulate, it kind of needs to pass that blood clot. And it can happen. So sometimes we have a double ovulation, but it needs to pass there. So that's why, for example, if we have a double ovulation on 2 ovaries, we have a higher chance to have 2 embryos than when we have a double ovulation on 1 ovary. And then the other thing is that we don't really have drugs that do work well for superovulation. One drug that we could potentially use is what we call recombinant FSH. However, at the moment, it is not on the market, so we don't have anything to successfully superovulate.
And even with the recombinant FSH, you know, I mean, ultimately we're talking about 1 or 2, we're never talking about 20 embryos. No. So I think you answered that question really, really well, you know, that there is just huge anatomical differences between cattle and horses, really. Another question that has come in for ovum pickup, can you do that when the mare dies? So let's say a mare breaks a leg. Can you aspirate her follicles and use those for in vitro fertilization for ICSI?
Yes, we can. We can do it, and we have done it for some of our clients. I think we've done 2 here now, which one produced 2 embryos and the other one as well. And basically, the successes are the highest in acute cases. So where a fracture occurs or something, a short illness, because you need to see those ovaries have a blood supply. So if a mare is chronically ill, they will have and receives plenty of medication that will go through her bloodstream as well. So like a fracture or something acute is more likely to be successful. And what I do advise is that if you can take CM swaps, and you can do them for if you can postpone it for a day, then you have your results of your CM swaps, because mares needs to have export papers to leave our country, and until we have a successful ICSI lab within the UK, you will need to have export papers. So and you can do CM swaps through PCR, so you'll have the results quite fast, and you have EVA and EIA, not EVA. You need EIA results, but those are things to think in mind. And as well, keep a hair sample with, because that's the thing. You need to be, sometimes certain stud books will ask you to prove the DNA of the foal after it has been born through embryo transfer. So I do advise to keep a hair sample with roots as well, just to kind of prove that later on.
Kaatje, thank you very much. Unfortunately, we are coming already up to 8:30. Those 1.5 hours have gone extremely quickly. We will try, we couldn't get through all the questions, and we will try to answer those even after this live seminar in the next few days, be it by email or on there. Thank you very much for submitting all. I would like to finish with one question is, you as a specialist in assisted reproductive techniques, you know, where is the future of equine reproduction in the next 5 years?
I think ovum pickup has picked up a lot, although, so I think for competition mares, we will do more and more ovum pickup because it's so easy to put a plan in. However, I don't think it will take the place completely from traditional breeding. Well, for me, traditional breeding, not completely, not natural breeding, but AI. I think people that want to have one-off foal, and it's going to be a lot of them carrying their own foals, that will continue. But I think the ovum pickup will grow a lot, and I think that will be a benefit for those female genetics as well, because although men believe that the male genetics are the most important, I think we're proving recently that that isn't the fact anymore, and ovum pickup will grow. And although, and some people ask me often about cloning and my opinion on that, but I think cloning is a bit standing still, and I think with breeding you want to go and make very interesting cross it, like and make something new and more exciting. And I think with ovum pickup and those things, we will. I think that is the future of breeding.
Wonderful, Kaatje. Thank you so